Good afternoon, good morning, good evening, whenever and wherever you are joining us from. Thank you so much for being with us. I'm Maggie Kuhl, the Vice President of Research Engagement at the Michael J. Fox Foundation. I'll be your moderator for today's webinar, A Year Like No Other in Parkinson's Research, our 2023 in review. With me today are three colleagues. Sohini Chowdhury is our Chief Program Officer. Thanks for being here with me, Sohini. And our co-chief scientific officers, Mark Frazier and Brian Fisk. As always, Mark, Brian, nice to chat with you as well. So,
always great to be here.
So today we are going to first introduce our, here we go, great, our patient-centered strategy. So the Michael J. Fox Foundation was founded by a patient. We are a patient-centered organization. So there's a lot on this slide, but really what we wanted to start off with was our commitment to centering people with Parkinson's at the heart of everything that we do. We learn about Parkinson's disease, the biology, the clinical experience, and that directs our strategy toward better trials, better treatments, and better care. So today we're going to share across the spectrum things that the foundation staff are talking about, places where our donor-raised dollars are investing in helping improve quality of life, cure disease, and perhaps prevent the Parkinson's process from ever starting. So with that, we're going to kick off. We are going to cover so much today. There is so much progress that has happened in the last year. We are going to keep things at a little bit of a surface level, but we have included a lot of links in the resource list and in the slides, which you can download in the resource list. So if you would like to learn more about a specific topic, you can click through and read or watch another webinar even on your own time. Or if we get to the Q&A section and you haven't heard enough about something, please bring it up at that point. So that brings me to our Q&A, which is at the center of your screen, the Q&A box. Please drop any questions throughout the hour in there. Our staff will be answering, or we're going to try and save a good portion of the hour that we have together toward answering as many questions as you can. Another housekeeping item, if you would like to turn on your closed captioning, there is a small CC button at the bottom of your screen, and that should put on captions in English. If you are watching us on demand, this will be available to watch anytime on demand. There's a number of other languages available as well. So with that, why don't we move into our first topic? We would be remiss to start any Parkinson's Research Year in Review without talking about the Biomarker Breakthrough. There are a number of very deep dives. We did a previous webinar. We've done a whole newsletter on this. And so we wanted to just give a little bit of background. Mark, why don't you remind us, what is the alpha-synuclein seeding amplification assay and why is it so important for research?
Sure. Thanks, Maggie, and thanks for having me today. This alpha-synuclein seeding amplification assay, or SAA for short, is a relatively new biomarker, and the foundation has been supporting efforts in biomarkers really for decades since we started in the early 2000s. And the reason we think this is so important is to have a more objective marker of diagnosis that can help doctors or neurologists in the diagnosis of the disease, and particularly early in the course of disease when the symptoms may be very slight. And until recently, this sticky protein alpha-synuclein, we've known for many, many years that it accumulates in the brains of people with Parkinson's disease. And there was really no way to measure alpha-synuclein until someone passes away and some pathologist looks at the brain under the microscope. But that has all changed recently in the development of a laboratory test. And this test actually was funded by the foundation, started in a university, and now has been spun out into a company. And this laboratory test is called the alpha-synuclein seed amplification assay. And very simply, what happens is that we take a drop of spinal fluid, put it into a test tube with normal alpha-synuclein. And in people with Parkinson's disease, this normal synuclein seems to seed amplification of clumped synuclein. And this clumped synuclein, after shaking for a day or two, can be visualized in using a fluorescence reader. And so this laboratory test is really exciting because about 90, maybe sometimes 93% of people with Parkinson's test positive for this seed amplification assay. So it's really the first time where we have a pretty accurate, objective laboratory test that can assist with the diagnosis of Parkinson's and detect this synuclein in a living human person.
It really is a groundbreaking discovery enabled by the contributions of many volunteers. So thank you so much if you've participated in one of the studies that have led to the discovery of this biomarker test. But thank you as well to the future volunteers who are going to help us improve it, because there is still much work to do in making this test more accessible and more useful for trials. And so there are some projects already in place, even well before this biomarker was announced in April of 2023. Our foundation was focused on improving its utility. And so, Sohini, maybe you can push us through to a couple of these next steps in this biomarker development.
Happy to, and hi everyone. Really pleased to join you today. So I guess there's sort of two levels of activities in terms of really bringing this breakthrough to the next level. On the sort of the first level of activity is really focused on sort of the technology around this. As Mark mentioned, this test at the moment is run on cerebral spinal fluid, which is collected through a lumbar puncture. And that is not the easiest procedure to go through. It's a very informative procedure, but certainly it limits the ability to be able to really use this test on a wide spectrum of individuals. And so one of the things that we're really focused on is on transforming this particular test into something that's more accessible, like a blood test, or, given the post-COVID world we live in, perhaps even a nasal swab. So there's a lot of work that's going on to try to translate this test from being run on cerebral spinal fluid into something that's more accessible and easier for individuals to undergo. A second technical component that we're looking at in terms of optimizing this particular test and improving it is really focused on making this test a quantifiable test. So at the moment, as Mark illustrated, this test tells you whether you do or do not have that alpha-synuclein protein clumping of interest. What it doesn't tell you is, do you have a lot of it? Do you have very little of it? Is it changing over time, etc.? And so the second sort of technical kind of work that we're really focused on is to try to make this less of a yes-no test and more of a test which could give us a sense of the spectrum of clumping that is happening and kind of correlate those that spectrum of clumping to what an individual is experiencing with the disease. And so those are kind of the two main buckets of activities that are focused on improving this test on a technical arena. But there's more that's happening, and this is really about ensuring that we're leveraging this test to its fullest when it comes to Parkinson's R&D and particularly Parkinson's therapeutic development. And as Mark mentioned, alpha-synuclein is a hallmark of the disease. It's the pathological hallmark of the disease. And so one way we're really focused on leveraging this breakthrough is really driving home the fact that this test should now be utilized in all clinical trials. Why? Because, well, if you're really looking to make sure, if you're really looking to test your therapeutic in a Parkinson's population, then you should make sure that that is the population that you have enrolled in your clinical trial. And the way to do that is to say, are the individuals enrolled, do they have this pathological hallmark of alpha-synuclein? And now we're able to test that through this SAA test. So we're really focused on making sure that as many trials as possible, where it is applicable, that they're utilizing this test to make sure that they really have the right people enrolled in the study so they can get the right sense of whether this drug or this therapy is really having the intended impact. The second way that we're really looking to kind of expand how we can utilize this test is that there have been some very interesting data that have come out of the development of this test that have shown that this test not only can detect alpha-synuclein in individuals with Parkinson's compared to those who don't, but interestingly, it's able to detect the presence of synuclein in individuals who do not yet have symptoms of the disease. So it may be a way of being able to identify people before symptoms start. And what that has allowed us to start to do is kind of begin to define Parkinson's disease, not just based on the symptoms that an individual may have. Do they have tremor? Do they have bradykinesia? Do they have gait or balance issues? But to actually define the disease based on, do you have this biological hallmark? Do you have other aspects of the biology, such as dopamine degeneration, et cetera? And the reason why this is so exciting is that it helps drug development makers be able to kind of understand what they need to target. Can they target a reduction of alpha-synuclein? Can they kind of prevent having synuclein and then go on to develop dopamine degeneration? Can they intervene so they stop that? And staging allows you to kind of have a framework by which you can orient your therapeutic development programs. And so that's really exciting. The last component is that Mark mentioned that about 93% of Parkinson's patients test positive on this test. That means that 7% don't. And that what we have also found out is that depending on a genetic mutation, you may actually have a higher chance of not being SAA positive, not being positive for this test. That begs the question of what else is going on. And so this starts to open up avenues for us to really understand what are the different biological processes that are under this umbrella of Parkinson's disease. And this is so exciting because for the first time, it really allows us to think about how do we get to a precision medicine approach for Parkinson's disease? How do we look at an individual and not say, oh, well, tremor's bad today, or this is the symptoms this individual has, but actually say, this is what's biologically going on in this person, and this therapy is targeting that biological process. So it allows us to really make sure that we're thinking about the right therapy for the right individual at the right time, and that matters. That's how you really make sure that a therapy is having the greatest impact for an individual.
You stole my transition. I had just written down right therapy, right person, right time. That's something that we say a lot at Fox, but it's true, and it's what we're going after. And to build on what you said as well, Sohini, what I heard too was we are running as far and as fast as we can with what we have, because we are. Oh, sorry, seems like someone's playing with the slides. So if you're a panelist and you're playing with the slides, please don't. Or is it just me? Is anyone else's slides going a little crazy on the end? No? Okay, I'm going to push us to number 5. I hope everyone's seeing number 5 that says more biomarker breakthroughs await. But as I was saying, we are running as far as fast as we can because we know patients are waiting, but we are also looking for the next improvement that's going to allow us to go even farther and even faster. And as you said, there's a lot of complex biology at play in Parkinson's. There may be people who alpha-synuclein doesn't resonate. There are things that are working together, and if the more that we can measure and understand, the better position we're going to be in. And so this alpha-synuclein SAA test is not the only biomarker that we are looking for by any means. And Mark, I wanted to perhaps toss to you to give just a highlight. These 3 are, again, by no means what we are looking at exclusively, but perhaps some of the things that people have heard about. So what's new with other biomarkers? What are we talking about at the foundation?
Yeah, I think this is just the beginning, Maggie, and you'll see a number of breakthroughs over the course of the next year or so. Already, we've seen some progress in an area to try to visualize and see the clumped alpha-synuclein in the brains of people with Parkinson's, and we've been funding this work for over a decade to try to develop neuroimaging approaches to really see a snapshot of synuclein in the brain. And this picture on the left is just one example of a study that was recently reported where they could see alpha-synuclein in the brains of people not with Parkinson's, but Parkinsonism called multiple systems atrophy. But we're really excited. There are multiple groups working on this, and we expect a number of different neuroimaging approaches to be tested in humans in the next year. So we're seeing a lot of progress, and that will only just complement the work of the SAA, because the SAA work, that laboratory test really doesn't tell you where alpha-synuclein is in the brain. And having these pictures really can show you the real estate, and then with a therapy show you if you can reduce the alpha-synuclein using these neuroimaging techniques. But as you both mentioned, there's a number of other biological pathways that we know are affected in Parkinson's. It's not just accumulation of alpha-synuclein in the brains of people with Parkinson's. So there's things like cell biology processes like lysosomal processes, which we like to call the sort of the garbage disposal for the cells. And we know mitochondria in the cell, which is like the powerhouse, the batteries of the cell. Those are affected in Parkinson's. And what's exciting is that there are now tools being developed to measure lysosomal activity or mitochondrial dysfunction in people living with Parkinson's. So we think there will be a number of different biologic markers that will be able to characterize this unique biology that's happening in individuals with Parkinson's, and ultimately target individuals with those precision medicine approaches that are targeting and going after the biology that is occurring in the individuals.
Yeah, and I want to call out too that it's not just for overall progression. Some of these markers are tied to the type of Parkinson's that you have, if you will go on to develop cognitive changes, or if you will progress at a faster or slower rate. And so back to Sohini's comment about right therapy, right person, right time. And so some of this work extends not only to slowing or stopping disease, but for people who have been living with PD for a while and giving them the best options to manage their disease. Which brings us to a question that we get very often, which a lot of you sent in during your registration, which is, what is coming to market soon? What should I know about? So the good news is that there are a lot. So Brian, maybe I'll toss to you to give an overview of what might be on pharmacy shelves in the next few years.
Sure. And it's, so we, talked at the beginning when you introduced the topic today about sort of the different stages of Parkinson's disease, and the sort of, from the early risk to kind of the early stages to kind of the advancing disease. And I think what's exciting is what we're seeing really in the later sort of stage pipeline, and actually what's, some of these cases actually being up for FDA approval are treatments that we think can really help those, people at the later stages of disease, where you're dealing with some of the, the complications of the existing medications and trying to increase your quality of life throughout the day. And so there's a number of approaches here. A lot of these focus on kind of a common theme that we've obviously had for many years in Parkinson's around the impact of giving people back dopamine into their brain, which is the loss of dopamine cells in the brains of people with Parkinson's is what really drives at least the movement problems that people suffer. And so these approaches are really, a sort of novel twist on how we can sort of bring that dopamine into the brain in sort of more optimal ways, in ways that maybe can kind of reduce some of the complications that can come with these types of treatments. And so a few of these groups, so AbbVie has an approach, ABBV951. A lot of these are named by sort of numbers right now until they come up with the incomprehensible brand names that we'll hear about later. But for now, ABBV951, Indio 612. These are two approaches that are looking to deliver dopamine medications through pump delivery. So something that would be sort of worn on the belt and sort of infused through the skin, basically, to try to deliver these dopamines in sort of a dopamine medicine in a kind of more continuous way throughout the day. And some of the trial results have looked good. These usually help people. They increase the amount of sort of good on time they have throughout the day, reduce some of the bad off time people might struggle with. So just, again, better ways to sort of improve those treatments. There's another type of dopamine approach called it's apomorphine, so it's not levodopa, but it's also similarly to being delivered by a pump type of approach that's by another
Maggie, while we get Brian back, I was just going to say, a lot of these approaches are engineering approaches. So they're repurposing existing approved drugs, but engineering them in a way that it makes the delivery more efficient or quicker and reduces side effects. And it's been remarkable to me to see the evolution of this field, because I would say 15 years ago, you talked to people in drug development, and they really thought that delivery wasn't an issue, and having Sinemet and levodopa was available. It was generic, and there really wasn't an unmet need. And I think it was through efforts of the foundation and others to really advocate and educate drug developers that there are significant side effects and issues with some of the existing dopamine approaches, and seeing these engineering approaches that are delivering it in a better way to reduce side effects and improve the
patient need, as we were saying at the top, listening to what matters to people with PD and responding our strategy. Brian, welcome back. Welcome back to the party.
Well, we're trying to cure Parkinson's. So apologies for the
exactly. That's your donor raised dollars are going to science and not to the Wi-Fi at the office.
So I don't know where I cut off, but let me.
Mark picked up beautifully. We were talking about engineering strategies and listening to the participant patients and study participants about their needs and filling a gap in care. I wanted to ask one more question on this topic. So Hini, for you, it's great that new drugs are coming to the market. How do people with Parkinson's disease make sure that their doctors are aware of these new treatments so that they can have them and have access to them? What is that link between approval, your doctor can actually prescribe it, they're aware?
Yeah, so it is actually more complicated than just getting FDA approval. And while I won't go into all of the steps, I think one of the biggest things, and all of you who are on this, listening to this webinar right now, will is we reach out and we try to understand what is going on with the reimbursement process. Are there attempts being made? What is the state of it, etc. Sometimes there's a lag, and that's kind of just the nature of sort of all of the steps involved. Sometimes it's set up beautifully so that once there's FDA approval and it's actually being marketed and it's available, reimbursements are already covered. But I think the most important thing is to have a frank conversation with your doctor. Many of you probably see movement disorder specialists who are at the forefront of really knowing about a lot of these new drug developments, and often they're running some of the trials that are testing the efficacy of these therapies, so they're very familiar. But many of you may not be seeing movement disorder specialists, and your doctors may not be as familiar with some of these new therapies that are coming out. And so I think you have a role to play in also being an advocate as you hear about this and asking your doctor to find out more, to read the literature, to direct them perhaps to information that maybe we have on our website or others may have, so that you can really have a proper conversation about which of these potentially new
less close to market, but still moving through the pipeline and development because, again, we know that these are the priorities of the Parkinson's community. So, Brian, let's try this again. Don't make any sudden movements, but maybe you can kick us off. We had a really exciting stem cell trial report earlier this year that you and I did a podcast on, which is linked in the PowerPoint slides there that you can download from the resource list if you want. But why don't you recap that conversation? Sure. What was the news that we shared?
Sure. No, and it's exciting. So, I mean, I think the idea of stem cells have been around for, probably a couple of decades now, and there's always been a lot of, I think, excitement and maybe even a little bit of hype around them. But I think the recent news was great because this was a company, Blue Rock Therapeutics. They've developed a stem cell replacement therapy. And so I kind of use that word specifically because I think when we talk about stem cells, sometimes it can get confusing. There's sort of stem cell approaches that are trying to sort of provide protective factors. And we'll talk about maybe another one in a moment. Then there are stem cell approaches that are trying to actually create new dopamine cells that can be transplanted back into the brains of people with Parkinson's. And so in this case, we're talking about Blue Rock is developing a stem cell replacement therapy. And what they did, it was an early phase 1 study, so it was only in a handful of people, but it was really one of, I think, the first times we've seen a true stem cell, derived dopamine transplant approach tested in Parkinson's disease and in a good sort of well done study. And they did it over about a year period in a handful of people. And I think the good news is it was safe. So that was, of course, question number one they wanted to ask and make sure that delivering these types of approaches into the brain, they're transplanted through a brain surgery approach, we're going to be safe. But they also did some imaging just to see if the cells, were still present. And so they could get any evidence that the cells were there and sort of, doing what presumably dopamine cells should be doing in the brain. And they, the data are looking good. It looks like the cells are there and that they're actually functioning as they should. So far, so good. It was good, certainly news for the company and for us to kind of hear the outcomes of this. And companies now, working on plans for the next sort of a larger trial to start testing this forward. And, I think it's just good news for the field because I think there are other approaches that are approaching the clinic as well that are also going to be delivering different types of stem cell replacement approaches. And I think this is always good news when kind of the first sort of companies step into this space and try it out and, get some of the initial data. It really kind of opens the floodgates for others to move into this space. So my guess is we're going to see more companies and more approaches like this moving to the clinic. It's, to be clear, this isn't a cure necessarily for the disease. The idea of what these treatments in their current form are doing is just putting, some dopamine cells back into the brain that will presume you still have to take your dopamine pills for the most part, at least most people probably will. And, that'll be converted into dopamine in the brain. And what these cells are doing is just giving you more of that sort of a dopamine sort of factory back in the brain so that you can make a more dopamine and hopefully get the benefits on your movement symptoms. But it's not a cure in the sense that we're not stopping the disease process. We're not somehow, changing the underlying Parkinson's disease itself. But if we can get people back a little bit of what they've lost, I think that'll be really important, especially, again, as we talked about at the start of the call today, for people, especially in the more advancing stages of the disease when the maybe traditional, types of protective treatments may not necessarily, be as beneficial because the disease has already progressed to a certain point. These types of approaches then become even more important to offer to people.
Yes, I think the words restore function, people are really happy to hear those, I'm sure. I'm going to move on down to walking and balance issues. We know that this is an unmet need and a lot of the available therapies, even though you think of them as movement symptoms, don't actually do as well with walking and balance problems, which are very challenging for independence and socialization and confidence in moving through our world. So we launched a funding program earlier this year focused specifically on better understanding the mechanisms and mechanisms behind and measurement and treatment of walking and balance issues. And we had more than 100 applications, which was extremely promising. We're already funding some very exciting work around this. So Heaney, tell me, why do you think there was such a big response? I think 100 plus applications for a sort of narrow focus RFA or request for applications for funding is pretty high.
Yeah, it is. And I think it's also exciting to see, because I think if you were to ask people living with Parkinson's or those around them, freezing of gait, sort of gait issues or balance dysfunction issues, these are going to happen to everyone is going to experience it. Usually towards the later, having had the disease for a longer period of time, but not exclusively. And certainly the worries that come about when you, as you articulated, when you're worried about being able to move with confidence and to have that sense of security that you can move about, that is limiting if you don't have that. And so I think what's especially exciting about this particular, the response to this particular funding program is that there's a recognition in the scientific community about what a huge unmet need this is for patients. And I think that is very, very exciting, because there is a lot that can be done in this area. There's a lot in terms of understanding the basic sort of biology and pathophysiology that underpins this. It's not about sort of dopamine. Otherwise, the medications we have would be helping. This is about something else, and what is it, and trying to get an understanding of that. But importantly, what can we do to try to address it, both in terms of pharmacological approaches, taking medicine, or non-pharmacological approaches, things that may help with innovative technologies that may help address some of the gait imbalance issues. And so seeing the response we got was really, it was rewarding, and it was exciting, because it's recognition by the scientific community that this is a huge unmet need, and there's a lot that still needs to be done.
I agree. And I would just add, I think it's a reflection of where the science is and our understanding of what causes these gait and balance issues. And there's been a lot of progress in understanding the brain and the areas that control walking and gait and balance. So I think the field has progressed, and that's why we're seeing a lot more research in this area.
And I think that's a great segue, if I may, Maggie, what Mark just said. You mentioned here about spinal stimulation. And as we've actually learned more about this, the science around this, we've actually been able to leverage advances made in other fields and see whether that can be applied to Parkinson's. And this spinal stimulation project that's listed on this screen is a great example of that, where a team has actually been able to restore walking in individuals who have had paralysis due to spinal cord injury. And their hypothesis is that the mechanism by which they were able to do this could hold true for Parkinson's as well. And so the foundation is funding this study. It's a small scale study to really validate, because they've already done this in 2 individuals, but to validate this in more individuals and really see whether spinal cord stimulation could address some of these gait and balance issues. And so these things can only happen as we have a better understanding of the science, as Mark articulated. We're able to see what's going on in other fields, and could that help us in Parkinson's?
Yeah, I remember from my graduate school days, we talked a lot about how, you tend to think the brain is where everything happens, but the spinal cord, actually, there's quite a bit of circuitry in there that if you stimulate in the right way, it can actually activate some of the basic things that we do, like walking, some of these basic behaviors that we have. So to see this kind of technology applied to that concept, I think is really exciting to see, and that might actually then have benefits for people with Parkinson's.
Absolutely. I want to take a quick detour here, because listening to you describe the challenges and the opportunities in treating something like walking and balance brought to mind that we received a lot of questions that started with how. How do I adjust to balance issues? How do I know what treatment's best for me? How do I get my husband motivated, who has Parkinson's and is experiencing apathy? And so I wanted to make a quick plug for our Buddy Network, which is a great resource if you or your loved one is living with Parkinson's disease, to gain information like ones that we're sharing, more about studies and research, but also just about how you navigate Parkinson's disease and how others are doing that. And so a lot of this, especially walking and balance, I've heard so many anecdotes of different cueing techniques or different devices that people are using for assistance. So I think it's linked in the resource list. If not, you can Google Fox Foundation Buddy Network. But all of those how questions, and while we're working toward better treatments that are randomized trial proven. There's also just a network out there that can help you navigate life with PD. So please check that out if you're not already. In the essence of time, I'm not going to go too deep into our last 3 bullets here, but I did want to call out that we know that there are so many other aspects of life with PD. These symptoms that we have listed here are some of the ones that we most frequently hear. As Sohini was saying, really challenging, really limiting confidence, independence, affecting quality of life. So we have a number of projects that are using everything from repurposed drugs to apps to spinal stimulation to try and uncover exactly how we can improve quality of life while we work toward therapies that will not require all of these different approaches and hopefully will treat Parkinson's overall. And with that, I think we'll transition to our next part of the conversation, which is cures. And I always put an S because I think the theme of our talk here is that it's not going to be one cure. It's going to be many really personalized for your own biology. So there is a lot in the pipeline. There's been some exciting progress this year. Brian, why don't you give us an overview, perhaps just starting with some of these targets that people may have heard of. We've already talked about alpha-synuclein. I think people hopefully are pretty aware that inflammation is a concern in Parkinson's disease and any others that you'd want to mention.
Yeah, no. So, when we look at the, therapeutic pipeline for Parkinson's, and the team here spends a lot of their days kind of digging through that and trying to make sure we understand what's moving forward and what challenges exist. Obviously a big part of the therapeutic pipeline today are treatments that are targeting, again, this alpha-synuclein protein that we keep talking about. And now that we have this, additional biomarker tool, I think we're going to see that, tool be used and sort of integrated into a lot of these trials going forward to think about how do we really, again, match these therapies to the people who are hopefully most likely to benefit from them. But there's been, a lot of great work. Some of the leading sort of groups in the pipeline that have these synuclein targeted therapies. There's a company, Roche, that's working on an antibody approach. So this is an immunotherapy. Basically, they've created a synthetic antibody that can go in and sort of target the aggregated clumped aspects of the synuclein protein, and through that process sort of try to get rid of it, much like our antibodies normally do when we're sick. This is a way to kind of just give you the antibody directly, and then hopefully it'll kind of help remove that synuclein. So they've been working on this for a while. They presented some they had some initial results, a little bit ago from their trial that suggested, some possible benefits of the therapy. They've continued to kind of look at those individuals for a number of years now, and sort of had done some additional analyses, and they reported on some of this over the last couple of months, suggesting that they're continuing to see those benefits over time, at least on the kind of the movement scales that they're using in those trials. So, it's continuing to look potentially promising that targeting synuclein this way might have some potential benefits. Obviously, they need to do kind of a more, larger trial, a well-controlled trial to really to demonstrate that more fully. But at least the signals right now are looking potentially promising. But it's not the only, synuclein's not the only target in the pipeline. We see a lot of diverse other mechanisms being looked at as well. You mentioned inflammation. I think that's a really exciting area. The idea that maybe inflammation, whether it's causing Parkinson's or it might be contributing to the cell loss over time. I think scientists are still sort of debating that idea, but at a minimum, the idea that we could be targeting the inflammation process directly is a very powerful one in Parkinson's. And so we're seeing a lot of approaches looking at that. Some of them are using more traditional small molecules and trying to target specific components of the inflammation pathway to see if they can alter that process. Others are using other kinds of approaches, like we mentioned earlier about stem cell approaches. Stem cells seem to produce a lot of interesting factors that might offer different kinds of protection. Some of those might be growth factors that kind of act like fertilizer in the brain. Others might be factors that help sort of modulate the immune system. And so there's a trial actually ongoing that we've been supporting for the last couple of years that's using so-called mesenchymal stem cells. These are a type of stem cell that's found in a lot of different places in the body, but in particular in places like the bone marrow and things like that. And so this investigator is using that approach, infusing these stem cells into people with Parkinson's to see if they can sort of show benefit over time with this approach. It's nice because this investigator in particular is taking a very sort of like rigorous approach to this. There are a lot of efforts out there that are sort of offering these types of mesenchymal stem cell infusions to people with Parkinson's, often at a cost and sort of directly, direct to consumer type of approaches. There's very little data on whether those approaches are really, offering any benefit, and data isn't really being collected about long-term benefit of those approaches. So for us, it was really important to then work with a scientist who could really rigorously test this. So we're hopeful to have some results from that trial next year, and that will help us kind of inform whether these types of approaches might also offer some benefit. There's a ton of other stuff in the pipeline, and we're running out of time that we can't really get into. I think we'll probably provide some links to places where people can go and kind of look at that broader pipeline. But again, really trying to focus on these different sort of biological sort of aspects of the disease that we think are relevant. And as we talked about, repeatedly before, this idea we're at a point where we really can start using some of these measurement tools now to really try to match up people to these different mechanisms and these different drugs that are targeting those mechanisms. So I think the idea of this sort of precision medicine future, I think, is less a future, and it feels like more of a present now. So really exciting to see all this moving forward.
Yeah, I want to ask you, there's been some in the news, or people might have heard of, there's been some testing of drugs that are very well known around diabetes and weight loss, these GLP-1 receptor drugs. The brand names are Ozempic or Wegovy or Mounjaro, people may have heard of, but they've been in testing for some time for their impact on Parkinson's. So what are we watching around that, Ryan?
Yeah. And I'll back up for a second and just say, a lot of these approaches are sort of fall into a category of therapy that we tend to call repurposed drugs. So these are drugs that are generally approved for another indication. There's maybe some biological evidence or rationale to think that it might be beneficial in Parkinson's. And so people will then try to test these in people with Parkinson's to see if they might see that kind of benefit. And drugs that target this mechanism, GLP-1, are an example of that. There have been others historically, a number of other repurposed approaches. I think I saw in the question thread someone asking about Ambroxol, for example. That's another repurposed therapy that's being looked at for a different mechanism in Parkinson's, and there's some ongoing trials there. But GLP-1 is interesting. It's the GLP-1 itself and the sort of the receptors that it activates in the body are really involved mostly in sort of metabolism. They really help us sort of deal with blood sugar and modulate blood sugar in the body. So they have some really important metabolic effects, and that's kind of why they were have been originally approved for diabetes to help with blood sugar, and more recently with obesity, where it seems to be helping people to lose weight. But over the last several years, there had been some data to suggest that it might have some protective benefits in sort of laboratory models of Parkinson's disease. And so a few years ago, some researchers started looking at this. They used a different GLP-1 drug called exenatide that's been available, and did some initial trials. And actually, the foundation funded one of the phase 2 trials for one of these drugs a few years ago that suggested that there might be some effects, that these drugs might actually have some benefits potentially at least on the clinical measurements that those trials used around sort of movement and things like that. More recently, in the last couple of years, there have been a slew of other trials that have tested not just exenatide, but some of these other similar GLP-1 drugs. And in the last year in particular, there's been a flood of sort of at least initial reports from these trials, again, suggesting, not always consistently, so that's, I think, important to remember to be clear about. Not every trial is sort of seeing the exact same thing, so there's kind of something odd going on. But they're seeing some clinical benefits of these drugs in people with Parkinson's. And so there's this sort of, I think, interest in thinking about what these results actually mean. Are they actually signals of real benefit? Are they something else related to, again, these are drugs that alter metabolism and do all kinds of things. So we don't know for sure what that might be, how that might be influencing the body. But we're monitoring it. We're sort of excited to see where this goes. There's a large trial in the UK that should be reporting out next year with exenatide. We'll have, I think more data from that trial to inform where this is going.
All right. Well, Sohini dropped. She was also in the office. So the wandering connection. Yeah. So Mark, I maybe it's just going to be me and you, but that means that you get our last bullet on this slide, which is that we have a lot still to learn about Parkinson's. We have so many targets. We have hundreds and hundreds of targets. These big ones like alpha-synuclein inflammation, these genetic markers or something sort of semi-connected to a pathway that we are just identifying and learning about. But we know there's a lot of darkness that we need to shed more light into around what causes and drives Parkinson's. So we have a number of efforts. One of the biggest ones is called the Global Parkinson's Genetics Program, or GP2, which is in partnership with our partner, the Aligning Science Across Parkinson's, or ASAP initiative. It reported a really big finding this year. Mark, why don't you share that with us?
Yeah, I mean, really, Parkinson's has been studied mostly in a Western European Caucasian population, and it's really important, not just socially, but scientifically, to understand Parkinson's globally. We know it knows no bounds, and Parkinson's occurs all over the world. And the Global Parkinson's Genetic Program is an effort to collect DNA samples from individuals, particularly underrepresented people in Africa, Asia, South America, to understand the genetic contribution of to Parkinson's disease in these underrepresented populations. And it's relatively new program, and what's exciting is even just as it's beginning, there was a report just this year that in people of African descent, both in the US and in Africa, there's a particularly specific DNA change in the GBA gene that contributes to Parkinson's. It's actually a different DNA change than other GBA mutations that have been identified in other populations, but it really just shows that this role of the GBA gene and protein that is involved in this this garbage disposal process is really important in Parkinson's disease. So we expect more discoveries to come out of this program to really understand Parkinson's at a global level and the biology that's occurring in everyone with Parkinson's.
One of the, I think, also really powerful points about the, that GBA finding and people of African descent was, obviously, found a variant or a DNA change in a gene we sort of knew about from other studies, but the sort of how common that change was in people of African descent was much greater than it is in people in other ethnic populations. And so I think it just, again, reflects the idea that, even if we go out in the globe and find that we all share maybe similar biology, for some populations, that biology may be much more sort of relevant to their form of Parkinson's disease. And I think that's really important to get back to this concept of precision medicine. How do we offer groups of people the right, treatments that really would impact their lives? And so people in these populations, you might then want to really consider these types of treatments that focus on, again, that lysosome biology, that GBA biology that you alluded to, that would be more relevant to a group like that than it might be in other populations. So it's really exciting to see, these kinds of data come out and really, again, put genetics in kind of this global context, which is really, really important.
Absolutely. Very, very exciting finding. And as you said, Mark, more to come from GP2. Absolutely not the last, or even, perhaps a little bit of what you are to hear from that program. I wanted to go back to something that we had chatted about earlier, which is access to care and treatments and building an informed and educated Parkinson's community that can help us achieve our shared goals of better care today, better treatments for tomorrow, better access and coverage for all. So Sohini, there's a couple things on this slide. You've already referenced the policy program, so you can chat about the National Plan for Parkinson's Disease, which again, hopefully many on this call have already heard, and the PDIQ and U events, which we host, are a great opportunity, and there's a couple coming up in 2025 to mark on your calendar. So just quickly, before we can go to the Q&A, anything you want to highlight around these programs?
I would just say that if you are close to the locations that are mentioned here, please do consider attending a PDIQ plus U event. These are really meant to kind of talk about how to put together the right care team for your needs, and really gives a much broader sense of the research that's happening. But importantly, it also highlights and exposes attendees to the resources, the local resources in the community that can be leveraged as one goes through one's journey with Parkinson's Parkinson's disease. One thing I will just say about the National Plan for Parkinson's disease is that for those of you who are not aware, this is a no-cost legislation that was submitted to Congress. It enjoys a very significant bipartisan support. But what it's really about is is sort of mandating on a government scale, mandating the existence of a coalition of agencies, federal government agencies, key opinion leaders, Parkinson's advocates, patient representatives, et cetera, to really think about how the government can help support and accelerate efforts to make Parkinson's a thing of the past. And so right now it is going through its normal sort of machinations as it goes through the two houses. But at some point, we fully expect that it will be approved. And then we're really going to be looking for many of you to partner with us to really make this happen and to form something that will have a lasting impact on Parkinson's. And so with that, I'll leave it there. We have a lot of information on our website about it, but it's an extremely exciting effort.
Yes, please advocate and share with your legislators the importance of this program. So with that, we're going to transition to the Q&A. We received 700 questions before we even began today. I have seen the hundreds that you all are sending in. Sohini asked me how long I wanted for Q&A. I said 20 minutes. We have so much to share. We've got 9. We're going to cover as much as we can in 9 minutes, I promise you. So team, succinct, there's lots more on our website. Sohini, starting with the policy theme, where are we with movements to limit exposure to environmental risk factors that we know raise risk of Parkinson's disease?
So there has been advancement in 2 particular areas. One is trichloroethylene, or TCE for short. Many of you may have seen a pretty devastating report that came out about exposure to TCE at Camp Lejeune, I believe is how it's pronounced. And that really spurred a lot of activity in a positive way. And so the team has really been working on this, and chances are that it's going to, government's looking at this, and that there's a chance that this is going to be the use of it is going to be dramatically reduced or banned. The second area of interest is paraquat, which I know we've talked about for a number of years. The foundation has worked with other groups interested in this area and has met with officials at the EPA. And so there is the EPA has signaled that it is willing to relook at its decision in 2021, where they basically said, we have no concerns with paraquat. So they have they've opened the door to reconsidering that decision, and we consider that the first step in really trying to kind of move the bar when it comes to paraquat.
Right, amazing movement there, and things that we know should not be so widely used with their high likelihood of PD. Mark, how many years in advance, and I think I put that in quotes because I think that's sort of the answer to the question, can a alpha-synuclein SAA test detect Parkinson's disease?
Oh, that's a great question. We don't know the answer to it, but the data suggests that in terms of the temporal pattern, alpha-synuclein seems to change first before the dopamine neurons are lost, or at least before we can detect the dopamine degeneration using a dopamine imaging agent. And then subsequent to that, symptoms develop. So, we're supporting studies to understand how early that happens. Previous studies have suggested that some of the pathology can occur 5 to 10 and sometimes even 15 years prior to symptoms developing. So I would guess somewhere in that range, but we really need to generate additional data to understand how early that happens.
Mm-hmm. Yeah. So the disease is starting, and this is where I make a plug for our PPMI study, which is where a lot of the results around the SAA test are coming. My PPMI banner behind me, and it is recruiting anyone aged 18 or older in the U.S. There's lots of different ways to participate. It's linked in the resource list. Please help us achieve everything that we are saying that we all really need by joining PPMI. Brian, the glove, the Stanford glove. What is new with the Stanford glove?
I don't know if this is anything particularly new. So obviously this has been around for a few years, and I think people are excited by some of the reports that have come out. So basically this is a hand-worn glove. what it does is it sort of stimulates, with sort of vibration, the fingertips, and the idea behind it. We funded some of the early sort of, initial work to try to understand the.
There he goes. I need like a little like theme song anytime like Brian drops off. Okay. Mark, do you want to pick up? What do we know is happening?
Sure. I think what he was saying was by stimulating the fingers through these vibrations, it seems to reset the circuitry and at least anecdotally reduce some of the symptoms, including tremor and slowness or stiffness. As far as we know, there are clinical trials that are ongoing to test this more rigorously. As I mentioned, it was more anecdotal evidence of people wearing the glove that seemed to have reduced symptoms, but we really need controlled studies, including people that might wear a glove that may not receive the stimulation or a different stimulation to really control that placebo effect that we know is common in Parkinson's disease. So I think those studies are ongoing. Currently, it's not available, I think, to the population and really needs additional data to make that available.
Brian, I'm beginning to think that Mark has, planted some device so he can just finish all
he wants there only be one CSO, not two. I think that's what's happening. But but it sounds like you guys ended the again, more data.
Yes. There'll be more questions for you. Stay with us. But Mark, staying with you for a second, question on if we're using artificial intelligence for Parkinson's research.
Yeah, this is popular across, not just medical research, but research in general. But there's two main areas that we've supported AI in Parkinson's research. One is to analyze sensor data. So this is wearable data using smartwatches or smartphones that collects massive amounts of data and to try to identify patterns that might occur with Parkinson's disease in a living normal environment. We've supported studies that collect a lot of wearable data and then now have funded work to analyze the data using AI or machine learning approaches. The other is in molecular data. There's been a tremendous wave of really deep molecular data, genetic data, but also protein profiling data in fluids. There are now approaches to measure different metabolites in fluids, alongside complicated imaging data. And so we've supported some AI approaches really to combine different types of data sets and again, understand different subtypes of Parkinson's disease, try to identify individuals that might progress faster versus slower and what might predict that from occurring. So it's really an effort to combine a lot of large data sets and identifying patterns that humans in traditional statistical approaches would not recognize.
That's why we need lots of data and lots of people to participate in research. So I'm going to take a page from my own webinar mentor, Dave Iverson, and do a little around the room to end us. It's the end of a monumental year, a year like none other in Parkinson's research. What are you walking into 2024? Feeling, hoping, collecting? Mark, why don't we start with you?
Yeah, I mean, what I'm really excited about is not just the breakthrough, the biomarker breakthrough, and having these tools that measure and can detect Parkinson's biology earlier, but also the therapies that are developing in parallel with those tools and the biomarker tools. And it's really a convergence of having the measurements and also the therapies that can go hand in hand and make clinical trials smarter and more efficient and really hopefully slow the disease.
Sohini?
Yeah, I think when I think about what I'm walking into in 2024, I think I'm walking into it with a really open mind, because I think to what Mark said, the entire landscape for how we think about therapeutic development and how we think about Parkinson's disease has changed in 2023. All of a sudden, we can look beyond the symptoms and actually start to understand what's going on in an individual. And we're starting to recognize that what is going on in an individual is different person to person who's dealing with Parkinson's disease. And I think that opens up huge doors for us, both in terms of how we think about, to Brian's point, building on this concept of what precision medicine could be for Parkinson's, but also thinking about how we can leverage all of this knowledge that we've built over the past couple of decades and really refine our understanding of what's happening for each individual and how to intervene in the best possible way in the disease that that individual has. And so I don't know exactly what that means yet, but I feel that we are on a cusp of a total seismic change of how we're thinking about this in a way that is really going to resonate and create impact for people living with the disease.
And Brian, thanks for being such a champ and overcoming your difficulties. So the last word is yours.
Great. So what I'm looking forward to, one, hopefully more reliable internet in the future. But two, I mean, I'll just echo Mark and Sohini and just say, I think it's a really exciting time. This biomarker tool, we can't talk about it enough because it's it's something we've wanted for so long. We're going to see its imprints all over the therapeutic pipeline in the coming years. Look forward to next year. There are going to be probably some new approvals for at least some of these more advanced programs that we talked about. So look forward to that, but also look forward to progress in the pipeline as some of these other approaches start moving forward as well. So we're going to get a lot of data next year. We'll know, hopefully next year when we're doing this webinar, we'll be able to talk about those exciting updates as well.
Thank you all, and thank you all for attending. Please access the resources. We had so much to cover. You asked so many questions. We hope the foundation is a resource for you to become empowered, informed, and true partners in our shared pursuit. So thank you for joining us.